Signal transduction pathways involve reception, transduction, and response. This usually involves phosphorylation cascades and secondary messengers. The binding of a ligand can result in cell growth, secretion of molecules, gene expression, or opening of channels. So as we go here, again, we kind of look at this process as in three steps. We have reception, where the signaling molecule or the ligand binds to some receptor. The transduction pathway is happening within the cell, and there's going to be modification of proteins, which includes activation of enzymes and possibly phosphorylation, attaching of phosphate groups. And ultimately, we have a response. So sometimes that response is one thing happening, and sometimes there can be multiplicity where one receptor, one ligand can cause multiple responses. But examples of responses, we can change the shape or the movement of the cell. We can cause the cell to grow or divide. We can also change enzyme function within the cell, and we can also alter gene expression. So as we go here, a lot of the examples I'm going to hit later in the models that I show don't involve changing gene expression. But again, with this idea, we have reception, but then a lot of our responses can involve gene expression. And with this plant example, sometimes the receptor is inside the cell, and sometimes the receptor is embedded into the membrane, depending on the type of hormone or ligand. We can have these modification of relay proteins, and then we have our response. So typically when we're talking about changing gene expression, we're going to talk about steroid hormones. And steroid hormones are lipid-based, which means they can go through simple diffusion and pass straight through the lipid bilayer. The receptor for these steroid hormones are going to be typically associated with hormone receptor complexes that are transcription factors. And that transcription is going to, what we might say, turn on a gene, which transcription happens. We make our messenger RNA, which can go to a ribosome. We can do translation and create the protein, and that protein can alter cellular function. So another important point to note is that not every hormone or ligand binds and interacts with every single cell in the body. When we start thinking about multicellular organisms, these hormones are only going to bind lock and key to specific receptors, and they will not affect the function of every single cell. So in this case, with non-steroid or peptide hormones, a lot of times they're protein or peptide based, and these cannot get through the lipid bilayer on their own, so the receptor is often embedded into the outer plasma membrane. When we have this, we're going to have secondary messengers involved, and the intracellular domain of the receptor will change. And this secondary messenger is going to become activated. A lot of times we see it as adenylate or adenylyl cyclase, which our secondary messenger is cyclic AMP or CAMP. So we start with ATP and we get this secondary messenger, which then we talk about this cascade. So a lot of things are happening, and eventually we get this response where glycogen is broken down, which allows glucose to leave the cell and go to the blood. And again, this originates with epinephrine as the hormone, and the response is going to be glucose leaving the cell. So we often see this, and originally we have protein modifications. I want you guys to notice that. So a lot of times in this transduction pathway, again, we have the initial reception, the binding of epinephrine to a G protein receptor, and we activate the protein. We modify these proteins in order to make them active. Now, again, we have an inactive to an active, and this is a transforming of the molecule into a different molecule, but then inactive to active, inactive to active. So there's protein modifications taking place. We also have these phosphorylation cascades, and as you can kind of see this, it looks like a waterfall cascading down. And a lot of these are kinases. And when we deal with kinases, a lot of times those are enzymes that are associated with attaching phosphate groups, which then does change the function of the proteins because they go from an inactive to an active state. The other thing that's important to notice here is amplification. We can start with one hormone, one activation, and end up with 1 times 10 to the eighth actual glucose molecules as a result. So we can send one message, but then through this cascading process, we end up with an amplification of the response. So many more molecules get involved. So again, that last one was epinephrine, but glucagon, similar process here. So again, as we see this, sometimes we make models to make better visual mental representations of a process. There's always going to be some inaccuracy involved because like even in this, we don't have a nucleus in this model cell, but we're going to try and notice the signal transduction pathway, the phosphorylation cascade, and see the response. So as we go here, we have our glucagon non-steroid hormone. It's going to bind to this G protein receptor, and that's going to change the intracellular domain. This GDP interchanging with GTP is going to slide along the cell membrane, activate adenylyl cyclase, which then is going to take ATP and turn it into cyclic AMP. So here's our secondary messenger. And from here, we can start ultimately a cascade. We're activating different molecules as they go, and we begin this phosphorylation cascade. All of these are meant to represent ATP. And as we go through these next steps, there's a lot of phosphorylation or attaching of phosphate groups that are taking place in this process. And we have these hexagons here. This is stored glycogen. And as this process happens, the glycogen is broken down into individual glucose molecules, which will then cause the response, which would increase the blood sugar and remove stored glucose from the cells. So as we go through this, another idea to consider is that there's a lot of steps, which allows for a lot of control. Now we have glucose in the blood, and we want to send it to other cells to be used. So we take it from stored cells, like in your liver, and now we're putting it into the blood because you haven't eaten in a while, and we're going to send it to other cells that need the sugar so they can continue to generate ATP. So insulin hormone is going to reduce your blood sugar, and it's going to put the glucose into the cells. But again, this has a pathway as well. We have our target cell. The insulin is a non-steroid hormone, so the receptor is going to be buried in the outer cell membrane. But then as we have this binding of the ligand or the insulin hormone, we are going to change the shape of the intracellular domain of the receptor. This is a tyrosine kinase receptor, which is going to cause dimerization or pairing of the intracellular domain, which causes this autophosphorylation, where again, it's a kinase, so then we are phosphorylating here. And again, as we go through this, you don't need to know all these steps, but just it's meant to help you visualize this phosphorylation cascade and the complex signal transduction pathway that's taking place. So we have all of these inactive proteins becoming activated. We have phosphate groups being attached in order to modify the proteins. Here, again, in order to move this vesicle of channels and embed them in the outer cell membrane to get the glucose in, a lot of times we think about insulin opening glucose channels, but the channels actually get embedded into the membrane, and we have this little kinesin motor protein, which is going to walk the vesicle along the cytoskeleton. But all of that, again, the movement is caused through changing of phosphate groups. And we ultimately get our response where the blood sugar is reduced and it puts the glucose into the cells to be broken down through cellular respiration. Another example here, we have antidiuretic hormone or ADH, where we are right here. This is in your kidney, capillary in your kidney. This is the tubule, and this is a cell of the kidney tubule. Ultimately, this is filtering and reabsorbing your blood, but then as we go through here, this is going to ultimately lead to the bladder and outside the body. If antidiuretic hormone is present, we're going to pull this water back into our capillaries and ultimately back into our blood. instead of peeing out all that extra water. So it's an anti-diuretic. It's going to reabsorb water to the blood from the kidney tubule. But again, we have G protein receptors here. We've got adenylate cyclase. We're going to end up with cyclic AMP, and we've got aquaporin channels. But again, we have these kinases and all of that kind of stuff. So as we go here, we have our target cell, the ADH. It's a non-steroid hormone, again, because the receptor is on the cell membrane and not interior, and it's not going to the nucleus. But as we bind, we have the intracellular domain of the receptor changing shape, and then ultimately that's going to activate an enzyme which creates the secondary messenger cyclic AMP, which then again we have this relay of protein changes, which then embeds the aquaporin channels on the side of the tubule, which then can allow quick absorption, reabsorption of water through the cell and back to the capillary. Last one here that we're looking at an axon terminal, and a lot of times we've been talking about these long steps of signal transduction pathways, and sometimes ligands just open channels straight away once they land because the receptor is associated with a channel. So as we have this depolarization of the flooding of sodium ions, we ultimately send calcium ions in at the end, which is going to cause this vesicle and neurotransmitters to merge and release the neurotransmitters, which is a ligand, and this is a ligand-gated channel, not a voltage-gated channel. So as the ligand or the neurotransmitter binds, it's going to open the channel here, which then causes that initial positive flooding of sodium ion channels, which can create the action potential, which causes more voltage-gated channels to continue sending the message. So again, as we go here, we've got reception, transduction, and response. We often are associating that with phosphorylation cascades and secondary messengers, and that ligand, that reception of the ligand can cause cell growth, secretion of molecules, gene expression, or opening of channels.